Welcome to the citric acid cycle experience. There are four main objectives of this experience. The first is to understand how carbon and hydrogens are used to generate ATP. The next is to know the names of the molecules that carry electrons to the electron transport chain. We also want to know how many ATP are generated by the citric acid cycle, and we want to see how oxaloacetate is reused in the citric acid cycle. Overall, the point of this experience is to visually see how molecules pass through the citric acid cycle to generate ATP. Here on the screen is an image of the citric acid cycle. We spend a lot of time memorizing this cycle and the names of everything, but we don't always see what's really happening. So remember, the end product of glycolysis or beta oxidation is the production of acetyl-CoA. So no matter whether we use fat or carbohydrate for energy, we are going to end up starting with acetyl-CoA. The main point of this cycle is to move hydrogen electrons off of carbon molecules. The carbon will come from the acetyl-CoA that we start the cycle with. We're going to move the electrons or hydrogen molecules onto another molecule called NAD+, and then transport those electrons to the electron transport chain, where it will generate the ATP. So let's go ahead and get started. Today in our examples, gray will represent carbon, red will represent oxygen, and white will represent hydrogen. There are a bunch of other colors, but these are the three that we will focus on. For this first reaction, we're going to use a starter molecule known as oxaloacetate to carry two of the carbons from acetyl-CoA around the cycle. We are going to use this enzyme, citrate synthase, to combine them. Go ahead and grab a molecule of oxaloacetate and the acetyl-CoA. We will also need some water, so go ahead and add that too. You can see how we added the carbons from the acetyl-CoA and the oxygen from the water to this molecule, and now we have citrate. We also were left with a CoA-SH, and we're going to move this out of the way for now, but we'll see it again later. So now we're going to keep moving our carbons through the system. Would you like to replay this section? Our next stop is the enzyme aconitase. Aconitase is helpful because citrate isn't in the exact shape that we need, so we need to change a few things around. Go ahead and put the citrate into the enzyme. Now we have a different shaped molecule that we call isocitrate. Isocitrate is in the right shape for our next reaction, which will happen by using the enzyme isocitrate dehydrogenase. Would you like to replay this section? Now, anytime you hear the word dehydrogenase in an enzyme, it is taking hydrogens off to transport electrons. This is the first step in the cycle where we are going to generate energy. We do that by getting rid of a carbon and moving a hydrogen onto one of our carrier molecules. Go ahead and put the isocitrate into the enzyme. The carrier molecule we are going to use is called NAD+. Notice the plus, that means that the molecule can hold an electron or a negative charge, so we can remove a hydrogen and donate it to NAD+. Add the NAD+ to the enzyme to watch the reaction. The NADH is going to leave and head to the electron transport chain. Let's keep track of this on our screen, and we will show the electron transport chain reaction in a second. Also, the use of NAD+ is a key point to remember for future quizzes and exams. The carbon dioxide is also going to leave as we breathe out, and that leaves us with alpha-ketoglutarate, which is similar to isocitrate, but it's missing one carbon. One other interesting thing about this step is that if we have a lot of NADH formed and the electron transport chain has a line building up to use it, the backup of NADH will stop this cycle from running, at this point since there won't be any NAD plus available. That's why we sometimes say this is a regulatory step. It just means things get backed up so it can't move forward anymore. This is an important point that you will likely be required to know on an exam. Would you like to replay this section? Now, the alpha-ketoglutarate we are left with will move forward to the next enzyme in the chain called alpha-ketoglutarate dehydrogenase. This step is very similar to the last. We're going to lose a carbon as carbon dioxide and then get an electron over to NAD+ to create another NADH. So let's go ahead and put our alpha-ketoglutarate into the enzyme. We're also going to put in the NAD+ so that we can get to the NADH. Now, since we're losing a lot of carbons, we're also going to be bringing back our CoA SH molecule and attaching it here. Now let's watch a few things happen. First, we're going to lose a carbon again. That leaves us with only 4 carbons. So again, we lose a carbon, which leaves us with succinyl. The carbon leaves as CO2, and we move its hydrogen to make another NADH that can move to the electron transport chain. Let's mark this on our tally. But we also brought our CoA molecule back in to hold onto the succinyl so it doesn't get lost. Would you like to replay this section? So, the newly formed molecule is called succinyl-CoA. Because we added this leftover CoA complex from our first reaction, we can do something interesting. We can remove that complex again and use one of the phosphate groups from it to generate a GTP molecule. GTP sounds a lot like ATP, and that's because in terms of energy, it is. It is used to hold a phosphate to directly make an ATP molecule without using the electron transport chain. So, let's see this reaction. The reaction is first going to need our succinyl-CoA molecule. Then we'll also need the free-floating phosphate and the GDP molecule. So add those into the enzyme now. Knowing how GTP can directly make ATP is likely something that will come up on an exam. So you can see we are left with succinate without the CoA now, and we created a GDP. Let's tally that on our screen for when we talk about our ATP production. Would you like to replay this section? Our next stop is succinic dehydrogenase. Now, our succinate molecule has some electrons that we can still take off and use in the electron transport chain. This time, though, we're going to use another molecule called FAD. This molecule is similar to NAD+. It holds our hydrogen electrons, but is going to enter the electron transport chain at a different spot, so it will generate a different amount of ATP. Let's add the succinate and FAD to the succinic dehydrogenase enzyme. FAD is another molecule that you are likely to be asked about on a test. FAD is going to carry 2 hydrogens to the electron transport chain, so let's tally that on our screen for later. Now that we are missing 2 hydrogens, our molecule is now called fumarate. Would you like to replay this section? To get a bit more energy out of this molecule, we're going to do a cool trick by using water to get 2 more hydrogens back. If we put fumarate and water into our fumarase enzyme, we can combine them together. Add them now. This new molecule is called malate. Would you like to repeat this section? Malate is going to be used by the next enzyme, malate dehydrogenase. For this last reaction, we're going to take two of the hydrogens we just added to malate and put one of them onto NAD+. Put malate and NAD+ into the malate dehydrogenase enzyme now. Now our malate will be called oxaloacetate because it's missing the hydrogen. And if you remember back to the start, we first combined oxaloacetate and acetyl-CoA together to make citrate. So we have come full circle. This is something you will want to know for future exams. Let's add one more NADH to our tally before we move on. Would you like to replay this section? Now, remember, we did all of this to be able to bring electrons to the electron transport chain. This chain works like a series of turbines. As the electron passes along the chain, it will cause free-floating phosphates to be added onto an ADP molecule to make ATP. Before we start, let's take a look at what we made during the citric acid cycle on the screen. We made one GTP, 3 NADH molecules, 1 FADH2 molecule, and 2 molecules of CO2. We already saw the 2 CO2 molecules leave during the cycle. Remember, those carbons came from the acetyl-CoA that we used to start the cycle. The GTP is going to give its phosphate over to an ADP to make our first ATP. Remember, this doesn't require the electron transport chain. So, let's begin the electron transport chain by clicking the button, and you'll see that we already have one ATP produced. Now, the 3 NADHs are going to start at the beginning of the electron transport chain, and they will each generate about 3 ATP. Finally, the FADH2 will go along the chain, but it starts at a slightly forward spot, so it's only going to generate about 2 ATP instead of 3. Knowing that NADH enters the electron transport chain earlier on and FADH2 enters a little bit later is likely to be a question on an exam. It also helps us to understand why NADH produces more ATP than FADH2. Finally, the hydrogens will pass back through the last enzyme on the electron transport chain and force a phosphate to connect to ADP, creating our ATP molecule. The next thing to remember is that for each glucose molecule we start with, we actually will create 2 acetyl-CoA molecules. So we need to double the amount of ATP produced on our screen, since the citric acid cycle will actually happen twice. So we can see that running the citric acid cycle will generate about 24 ATP. Remembering that this cycle creates 24 ATP for each molecule of glucose is an important number to remember for your exam. Now think back to our 4 main objectives of this experience. We wanted to understand how carbon and hydrogens are used to generate ATP. We wanted to know the names of the molecules that carry electrons to the electron transport chain. We wanted to know how many ATP are generated by the citric acid cycle, and we wanted to see how oxaloacetate is reused in the citric acid cycle. You should now understand how acetyl-CoA is donating carbons to start the cycle. We are taking hydrogens off of these carbons and giving them to NAD+ and FAD to take to the electron transport chain. We can generate about 24 ATP per glucose molecule that is oxidized, and we can see how we end up with oxaloacetate at the end of the cycle to be reused with another acetyl-CoA. If you'd like to repeat any steps of this content, feel free. Otherwise, let the research assistant know that you're done with the experience. Thanks.